going to start recording, and I believe the recording is on. So for those of you, be aware, if your camera's on, you will be recorded in the corner of the screen. Thanks, Heather. I've got 21 participants. I might give us one more minute here, Jacob, before I give this start. You don't go by Jake, do you?
Not normally.
Or Jakey baby, or
I won't be
sorry.
Sorry.
Jake is my grad student.
What's that?
I said Jake is my grad student who I unfortunately invited to the meeting. I apologize for that.
Oh, if he slips in the back, it'll be all right. In particular, if it's something he's very interested in, but typically we invite the postdocs and academic staff.
Yeah. Okay.
Yeah. Okay. It is now 3:33, so I might just sort of start by segueing into my introduction as people still continue to drop by. It is my great pleasure to introduce today's chair's lecture speaker, who is one of the freshest faces in the Department of Biological Sciences, Dr. JacobBerry, who arrived here. We were just chatting a bit. I guess Jacob arrived just inside of a year ago. So in the post-COVID era, Jacob and Anna arrived from the US and crossed the border and quarantined and did all that stuff, all those fond memories of arriving here, right? Well, here we are, a year later since the start of the pandemic, and we're still locked down and we're doing this remotely, but I'm really pleased that everybody's here, and I'm especially pleased to introduce Jacob today. So a little bit of background about Jacob. Jacob hails from Texas. He did his BSc in physics with a computational option. I guess that means he did a lot of computationally kind of physics as an undergraduate at the University of Texas in Austin. I understand that Jacob took some biology courses while he was a physics major, and that kind of switched him on to biology. In particular, he was quite drawn to the complexity inherent in neuroscience and in neurobiology, which he encountered as an undergraduate. So when he went on to graduate school, he did a PhD in developmental biology at Baylor, where he sort of really cut his teeth on fruit fly neurosciences. Following his PhD at Baylor, he moved to Florida to take on a postdoctoral fellow, and then that evolved into a senior scientist position at the Scripps Research Institute in Jupiter, Florida. He was there for quite a few years, until 2020, when we recruited him to come here to Alberta. He moved here as an assistant professor, as I said, in the spring of 2020. And I'd like to think that Jacob was drawn to Alberta, where the stars are bright and they're big at night, much like they are in the heart of Texas. And I understand that Jacob also spent some summers in Montana and Colorado growing up near the Rocky Mountains. And, we aren't that far from the Rocky Mountains here. So I think there's a few things that maybe appeal to Jacob beyond what an outstanding institution this is to be a biologist at. So with that, I will turn the floor over to Jacob, who's going to share with us an unforgettable talk about how fruit fly memory. Jacob, the floor is yours.
Thanks, Dave. Thank you for a very nice introduction. I also want to thank the Department of Biological Sciences for giving me a chance to present my work. And, with COVID and everything and restricted interactions, it's nice to be able to get to know some of you and to present my work and let you guys sort of know what I'm doing. And so basically today I'm going to talk about my research looking at the dopaminergic control of memory, both learning and forgetting. My model organism of choice is Drosophila. You can see in the bottom left. And so really the first portions of the talk will be sort of going over a series of stories that I've been a part of that are very interconnected, relating to dopamine and memory. And towards the end, I'll present some of my sort of more recent endeavor and pursuits and sort of a project that I'm working on currently here and starting in my new lab. Okay, so first, talk about memory in general. So brains store information about the world. So the point of this and sort of the function is to integrate sensory information with things like value or consequence about the environment, and through a process called learning to basically adapt your behavioral responses so that the animal organisms are more successful. And so looking at learning as a process, it's thought or it's been shown to involve changes in synapses, basically the cell-to-cell connections between the vast numbers of neurons in the brain. And even in a simple brain like the fruit fly, there are vast numbers of neurons and vast numbers of synapses. And so it's thought during learning that these synapses are changed, and they can be either potentiated or depressed. And so these changes, so the learning process itself is setting down a pattern of changes across the synaptic network. And so from an abstract level, these changes collectively are thought of as a memory engram. So sort of the set of changes that have occurred during learning that sort of constitute the foundation of memory. And neuroscientists have spent the majority of the time in the past, up until very recently, focused on this idea of learning and how this process is working. And while this is a very fascinating feature of the brain, it is not the whole story. Also, as we've sort of developed techniques to sort of look at the physiology of synapses in the brain, when scientists measure different aspects of synapses physiologically, we often refer to the changes we see after learning that are underlying the memory as memory traces. And I use that word later in the talk as well. But yeah, so learning is not the whole story. And I'd like to convey today that brains have also evolved the ability to forget. And so a lot of you may wonder why is it there are functional, sort of biological systems set up to forget. And so really that comes down to, I think, a couple of compelling reasons. One is very obvious, that there are finite memory resources or space in the brain. Even though there are a vast number of neurons and vast number of synaptic connections, if you think about what this picture is sort of illustrating is all the people that you've met in your life, for example, for decades of life, right after meeting each one of these people, you would remember a lot of information. You would be able to probably recall what color the shirt is they're wearing, aspects of their personality, and so on. All this information was stored at one point in the brain, but you're unlikely to remember all this information throughout your life, and nor probably is it beneficial to remember all this information. So I think the ability to forget allows to sort of a memory economics standpoint is good. So the other sort of, I think, compelling reason for why forgetting has evolved is to deal with an ever-changing world. So the world is not just static and completely the same all the time. What is true one day is not necessarily true the next day. And this is just a little example from Richards and Franklin review on forgetting in Neuron. And just an example of how if you have your favorite cafe that is sort of southwest of your house and you walk to it every day, you sort of ingrain that in your memory, but then one day they can actually relocate their store or the cafe, and your brain has to update and sort of learn the new location, and so you need that sort of cognitive flexibility, and it's no longer important to remember the old location. And so forgetting allows for cognitive flexibility. And the other, I think, compelling reason to forget is to remove inappropriate and harmful associations. And examples of this come to mind, like drug addiction, for example, and PTSD. And so this picture is an illustration of sort of drug addiction, the problem with drug addiction and relapse. And so here, a person that's using their drug of choice is using a lot of common neutral items like currency, in this case, dollar bills, a credit card. And the problem is they form an inappropriate association with these everyday objects, such that whenever they do try to quit, essentially quitting becomes essentially impossible because they end up going through relapse because they have these sort of inappropriate memories. that would definitely benefit them to be forgotten. And in the last decade or so, scientists in various different, studying various different animal species have found forgetting mechanisms, including in the worm, in insects like fruit fly, and then also in mammals. And so the neurobiology of forgetting can be studied with model organisms, and that is sort of one of the major focuses of my lab. And so, you know, I use fruit flies as a model system for a variety of reasons, and one of the major reasons is because they do display a large variety of robust behaviors, including associative memory, which is very relevant to other animal species. And so today, almost all of what I'm going to talk about revolves around a particular paradigm of associative memory, where the flies associate odors with a negative punishment like electric shock. And so to briefly sort of summarize this paradigm, it's pretty straightforward. Basically, there's a training phase. So we take a group of flies and we expose them to an odor that they have never smelled before. These are simple chemical odorants. And for example, illustrated in purple here, this odor is simultaneously paired with a series of punishing electric shocks. And then the animals are exposed to a second odor, which is like a counter odor that they've also never smelled before, which is not paired with the electric shock. And so we're asking the flies to essentially remember that this odor is bad, or this odor predicts punishment, rather. And so we can test how well they remember this by putting them in what we call a T-maze apparatus, where the flies are allowed to distribute for a few minutes between two different arms. One arm contains the odor that was paired with shock, and the other arm contains the counter odor, not paired with shock. And when we do that, we can essentially quantitate or measure their memory through basically a performance index based on the distribution of the flies between the two arms. And this is example data from wild-type flies, normal flies, and basically you can see that you can measure this memory at different time points after training to measure the memory retention. And right after training, pretty much the vast majority of the flies will robustly avoid the odor paired with electric shock, similar to what you see in this illustration here. However, afterwards, this memory decays quite rapidly and is forgotten. By one day or 24 hours later, there's really no measurable memory present or retrieved. And so basically, despite having a simple brain, the fruit flies are able to form robust memories and make them amenable to studying the neurobiology of memory. And the other major reason why Drosophila is a great model system is the fact that there's a real large cohort of neurogenetic tools that are constantly evolving and shared pretty easily across the community. And I'm going to sort of go over the sort of fundamental ones that will come up in my talk. And so the major one that is used for biologists in studying Drosophila is the Gal4-UAS system. And so this Gal4-UAS system is actually a yeast transcription factor system. So Gal4 is a yeast transcription factor that the flies, obviously, they do not have this Gal4 in their genome naturally. And so what we can do is make a transgenic fly line called a Gal4 line, which we insert a transgene in it into the genome that has this Gal4 transcription factor from yeast that is downstream of a tissue-specific promoter DNA containing enhancers and such. And essentially, this will allow the Gal4 to be expressed in specific cells and specific neurons. And but the Gal4 doesn't really do anything because the fly lacks the UAS sequence-based genes. And so the UAS is upstream activating sequences of DNA that the Gal4 would bind to and transcribe. So in addition, we can then make a UAS line of flies where we stick in another transgene that has our gene of interest, something we want to express in the fly, that is downstream of this upstream activating sequence for this yeast Gal4. And so in this fly, this gene of interest is not expressed because there is no Gal4 there. And so both of these fly lines will have normal phenotypes and normal memory. However, when you've made them together, you create flies that you get GAL4, so you have both transgenic elements there. And so you get the GAL4 expressed in specific neurons. And then that GAL4 causes transcription of your gene of interest. And so this is a way for us to express proteins of interest or tools or other things in specific neurons. And the community has developed GAL4 lines, thousands of lines. And these are just two examples. There's a lot of these lines, or some of these lines are very specific. And this is sort of an example of how specific. So the brain of the fly is in purple here, which is stained with a synaptic marker. So this is, all the purple is basically a synaptic neuropil. And you can see the green here is a GFP that's being expressed, a UAS GFP that is being expressed in a very specific GAL4, with this very specific GAL4 line in this specific circuit. And this is a separate circuit that's being expressed. And together, when you couple this with a large set of UAS-based tools that have been developed, for example, there's several very large UAS RNAi lines that allow us to knock down or disrupt the translation and production of sort of a majority of genes in the fly genome. Or we can express reporters of neuronal parameters like calcium reporters or cyclic AMP reporters, so that we can actually measure specific neurons' responses to stimuli and how they change with memory. And then also neuronal activity modulators, things that we can express in specific circuits and allow us to control these neurons, either increase their activity or decrease their activity. So altogether, with the robust behavioral phenotypes and in this set of neurogenetic tools, I think it's a really nice model system to really kind of dig into the mechanisms of memory. And so when we talk about memory, I think the first thing to really talk about is where the memories are stored in the fruit fly brain. And basically, if you look on the left, we have, once again, this outline here is the fruit fly brain. And in the center of the brain, or actually towards the anterior portion, you see this L-like structure called the mushroom body. And it's been known for many decades now that if you block the activity of these neurons or ablate, kill the neurons that make up this mushroom body structure, the flies are unable to learn. Lots of different types of associative memory are basically gone. So we know that this mushroom body structure is critical. It's basically a critical memory center in the fly. This is the mushroom body GAL4 line that drives in the mushroom body neurons. And so yes, this L-like structure is essentially the principal component of it are an array of mushroom body neurons. I have illustrated here on the right. And so these neurons have their cell bodies towards the back of the brain, and they receive sensory information through their dendrites, which are actually outside of this L-like structure. And then they project axons that bundle together to make this L-like lobe structure. And there are a couple of types of mushroom body neurons. I'm not going to get too much into that. For simplicity, I've shown one type that does not, the axons do not branch, so they just make this horizontal projection. But there are two other mushroom body neuron types that actually have a branched axonal projection. That's why there's an L-like shape to the lobes. But I think what's important to convey to you is that studies have shown that these mushroom body neurons respond to different odors in a very specific way. So when the animal encounters specific odors, you get a very sparse subset that are different between odors responding. So essentially, if you think of it as a mushroom body array as a network that is sort of encoding a sparse representation that represents each odor that they encounter. And so going forward, so we kind of have an idea of how the sensory information that for the associative memory is getting into this memory center. But how is the value or the consequence of, that's being associated with the sensory information, how is that getting to the brain? And that's where the dopamine neurons come in. And so this is a picture of the entire brain again in purple, stained in purple is the whole brain. And in green is GFP being expressed in a specific subset of dopamine neurons. A specific cluster called the TPL1 dopamine neurons. And they innervate specific parts of the mushroom body lobes. So you can see here, there's basically, they innervate there are 5 different types of these in this cluster of dopamine neurons, and they essentially send their axon, send their projections out. They receive, with their dendrites, receive the information from outside of the mushroom bodies, and then project their axons and innervate and make synapses into these non-overlapping compartments of the mushroom bodies. And what's been shown is that these dopamine neurons, in particular this class or this cluster of dopamine neurons, responds very strongly to punishing information like the electric shock, for example. And they also, if you block these dopamine neurons during the learning process, the flies are unable to form aversive olfactory memories. And so basically, the idea is that these dopamine neurons are conveying this shock information. And if you think about in terms of where the memory is stored, you can see that the mushroom body neuron synapses are where both the sensory information and the value or consequences are converging. And so the sensory information being represented by a specific and sparse set of mushroom body neurons being activated, and then the dopamine is sort of registering the consequence or value to be associated with that sensory information. And so when I first started studying this as a PhD student, I was interested in what else these dopamine neurons do. And I was particularly interested in how they may be affecting memory retention. And so what I wanted to do was to specifically manipulate this PPL1 dopamine neuron cluster that's involved in forming the aversive memory. I wanted to specifically manipulate them after learning has occurred. And so what I did is I set out to measure 3-hour memory. So that's a memory time point where the memory has decayed maybe 50% or so. So I trained the animals, and then I tried to manipulate the dopamine neurons between training and testing, so that they function normally during the training and testing, but you're changing the way they function in between. And so to do that, I blocked the output of these dopamine neurons using a thermogenetic tool called JacobBerry, which is a temperature-sensitive mutant of JacobBerry. And so you can express these in neurons, and it's a temperature-sensitive dynamine protein. And it essentially will shut down when you raise the temperature of the flies, because the flies are cold-blooded. So when you raise their environment temperature, the neurons match that, or they basically become the temperature of their environment. So you can essentially raise the temperature and shut down neurotransmission in a sort of reversible and rapid manner. And so when I did this, I found quite surprising result. Essentially, the memory was greatly, well, I should say the forgetting was greatly reduced. So where they almost retained all of the original memory, whereas the control animals had forgotten a good portion of the memory. And so this was quite surprising. And this indicated that the dopamine neurons are doing something other than just the learning, involved in just the learning. They're also regulating the rate of forgetting, and that after learning, they have some kind of output that I'm blocking here. But conversely, I also did the opposite experiment and overactivated these dopamine neurons. And so I used a temperature-sensitive cation channel called TRPA1. So if you raise the temperature, this channel causes calcium influx, and you end up activating and driving the neurons you express it in. So when I overactivate the dopamine neurons, or activate the dopamine neurons after training, I essentially obliterated the memory performance. So the flies basically behaving like they do not remember any training at all. So this was a kind of a foundational result for us, and really opened up our understanding of what the dopamine neurons are doing. And it looks like if you block the dopamine neurons, you slow down their output, you slow down the rate of forgetting, and if you increase their output, you can sort of accelerate forgetting. And I also narrowed this down using different drivers to at least these 3 dopamine neurons here, with the open circles. And so, this data indicated that the dopamine neurons had some kind of activity that was causing forgetting, and I wanted to see whether I could actually measure that activity. And so to do that, I used in vivo functional imaging. So I made a preparation where you can take a fly that's intact and awake and tether it to a platform. We cut a small little window in the top of the head cuticle, and you can position a fluorescent confocal microscope above this and perfuse saline to keep the fly healthy. And you can image the activity of neurons if you express reporters. And I expressed, in this case, GCAMP, which is a calcium reporter. And I expressed it specifically in these PPL and dopamine neurons. And so I wanted to look at, basically, I looked after training, and what I saw was, in fact, there was robust calcium transients in 2 of these dopamine neurons. So the ones with the filled in circles had really strong and ongoing activity, whereas the one that is not filled in here was quiescent. It had really no activity. And what was also interesting is these 2 dopamine neurons, they had basically a carbon copy pattern of activity, and it was also the same pattern that was going on if I looked at calcium signaling in the dendrites as well, which I think suggests that there's some kind of network information coming in that's driving both of these dopamine neurons and sort of releasing a forgetting signal, if you will, releasing dopamine after learning that's sort of degrading and removing memories with time. And so I then wanted to try to understand what is receiving this dopamine signal. So which dopamine receptor is responsible for these 2 different roles, really. And we knew and we know that there are 2 major dopamine receptors that are highly expressed in the mushroom body neurons. And one of them is DOP1R1 and DOP1R2. They're also referred to as DDA1 and DAM. Some of my figures have DAM and some have DOP1R2, for example. I apologize for that. But essentially what we knew is one of these dopamine receptors, DOP1R1, was critical for memory. So those flies that do not have this receptor, they do not learn at all. And so we knew that receptor is critical for forming the memory. However, not much was known about this DOP1R2. And so as you can see here, it's an antibody staining for DOP1R2. And you can see it's very specifically expressed in the mushroom bodies, indicating potentially a memory function. And whenever I measure their memory retention compared to flies that lack this receptor compared to a wild type, so the black is the mutants, and so flies mutant for this receptor, their memory is prolonged. So the retention is elevated, and so at 3 hours, it's almost double the memory is remaining. And then even remarkably, by 1 day, where normal flies have not retained any of their memory, these flies still have robust memory remaining, indicating that one receptor is involved in learning and the other receptor is involved in forgetting, leading to this sort of summary model here. So we have these 2 receptors. They're expressed in the mushroom body neurons where we think the memory is being stored in these synapses. And so during learning, the dopamine neuron is being activated by the electric shock to convey this value information and this signaling through this DOP1R1 to facilitate the formation of the memory. And then afterwards, there's this ongoing release of dopamine that is driving through DOP1R2 to essentially remove the memory over time. And since this study, other studies have confirmed this dual role for the dopamine neurons, including in other compartments in the mushroom bodies, which I can talk about a little bit later. In particular, like reward memory. So it even seems to be working in a similar way for reward memories, which are stored in a different part of the mushroom bodies. And so I don't have enough time to go into this story in too much detail, but I wanted to understand what was driving this ongoing activity in the dopamine neurons. And essentially, sorry, I was trying to pause that video. It's causing some lag, I think. But basically, I have another study where I looked at, by putting a fly on a ball like this and watching them walk around and recording the dopamine neuron activity when they're locomoting, when they're resting or sleeping, I basically found out that the dopamine neurons, the ongoing activity is regulated by the behavioral state of the animal, and the forgetting is regulated by the behavioral state. Such that if I did certain manipulations to increase the arousal of the animals or to increase their sleep or drive them to sleep, and essentially when the animal's aroused, you have more ongoing activity and a faster rate of forgetting. And if you make the animal sleep after learning, it preserves their memory by blocking this dopamine neuron activity. And so really it comes to a question of trying to understand now how the dopamine neurons are altering these mushroom body neuron synapses to encode the memory. So in other words, can we find a memory trace in these compartments of the mushroom bodies? And then how are these memory traces being altered during forgetting? And to really attack this question, I need to sort of talk about a little bit more of the circuitry that has been revealed since I started working on these, on the dopamine neurons. And so this is sort of to kind of understand. So I've been talking about how associative memory is, associating sensory information with some kind of value to lead to some kind of altered behavioral response. And I've shown that the odors themselves can activate mushroom bodies, so the sensory information comes through the mushroom body neurons. And at least the punishment value here, the negative values, seem to activate the dopamine neuron, these particular class of dopamine neurons, like I've talked about gamma 1, gamma 2 here, and there's also gamma 3 that I haven't talked about that also seems to respond to punishment. And what is revealed by a large effort, across the Drosophila neuroscience community, really started by the Geneva Research Campus. They essentially tried to uncover all of the neurons they could find that connects to the mushroom body lobes anatomically. And what they found is a lot of different things that are quite interesting and really kind of led to my next study, really, and how I approached it. But basically, one of the things they found is there are other dopamine neurons here, gamma 4 and gamma 5, for example, that innervate a different part of the axons here of this particular mushroom body neuron type, for example. And these respond to rewards, rewarding stimuli like food reward, water when they're thirsty. So rewarding information activates these neurons and seems to encode reward-associated memories in these parts of the axon. And so you get this extra branch here. So now we know how two types of sensory or value information are coming in. And so essentially the mushroom body neuron synapses are being segregated into distinct sort of value-like encoding components. But then the question then is, how would the mushroom body neurons, how does this information get converted into a behavioral response? And so this was a big question in the field until this big effort to really try to uncover what's connected to the mushroom bodies here. And what they found is there's another layer of neurons called the mushroom body output neuron, or MBOn for short. So these are called output neurons because they have their dendrites completely, or dendrites innervating the mushroom body neuron synapses here. And they project their axons out to take information outside of the mushroom bodies downstream. One thing that's interesting is that they're paired off, so they match off to each of the dopamine neuron types. So indicating that each of these dopamine neuron types seems to potentially regulate the synaptic connections to this to a distinct output neuron, and also that these output neurons are divided into two types or two classes. And they figured this out because by essentially activating these distinct output neurons in isolation. And so what they found is they activate these ones in blue, these output neurons in blue, they actually seem to drive like an approach-like behavior, indicating that maybe these output neurons are registering kind of a positive valence to the animal, and then the ones in purple do the opposite. They drive sort of an avoidance-like behavior if you activate them. And what's important is if you activate both of them together in the same fly, you essentially get neutral behavior or no behavioral response. And so this led to their model, or a model in the field rather, where the sensory information that's being, like the odor, is activating a mushroom body neuron, for example, or a set of mushroom body neurons. And then this information is branching into two pieces that drive two different alpha neuron components here. And that this information is integrated and balanced in such a way that if the animal essentially encounters an odor it's never smelled before and hasn't learned anything about, it'll have like a neutral response, because it'll be a balancing between these two components here, the two output neuron components. And then the dopamine neurons are sitting there at a location such that they can differentially alter those two components, right? So if you have some kind of, if you have a punishing memory formed, the punishment is only activating the dopamine neurons, these dopamine neurons that would be regulating the approach alpha neurons, but not activating these other dopamine neurons. And so you would be sort of shifting that balance towards aversion, for example, and vice versa for reward memory. And so I think it's a really elegant circuit, and I think it's quite fascinating. And you know, it's a simple brain, but it sort of illustrates how a simple brain can encode these different value information and form both repetitive and aversive memories. And so what I wanted to understand, because I've been studying this gamma-2 dopamine neuron and how it's forming memories and also involved in forgetting, I wanted to see if I could find a memory trace downstream of this gamma-2 dopamine neuron, meaning I should look at this gamma-2 alpha neuron and see if I can see if the synaptic connectivity to it is changing during learning. And so, yeah, so basically, yes, can we find a memory trace, you know, produced by this, encoded by this dopamine neuron? And then how does this memory trace change during forgetting? And to do this, I did some more in vivo imaging. Before I did that, we focused on this alpha neuron gamma-2 because it is the one that is paired off with this dopamine neuron here. And so we looked and we did some behavioral experiments to show that basically if we block this output neuron during short-term memory retrieval, it impairs it. So we knew this output neuron in this compartment was likely important for short-term memory retrieval. And also Anna Fan, also a new faculty here and my significant other, she collaborated with me on this, and I don't have time to show the data, but she essentially went in and did structured illumination microscopy and basically showed that indeed the mushroom body neuron array, or sort of the array of these mushroom body neurons, are synaptically connecting to this one output neuron here. And so what you have is all these different mushroom body neurons that respond to different sensory information, all converging on essentially a single, the dendritic field of a single output neuron here. And the dopamine neuron is positioned here to release dopamine and regulate these connections. And so I wanted to look at how this output neuron, its response to odors and stuff are changing before and after learning and forgetting. And so to do that, I expressed GCAMP in the output neuron so I could measure the calcium responses and its neuronal response to stimuli. And I made another preparation where the fly could, I could image, it's tethered to a platform with a hole in the head again, and so I can image the brain and image this output neuron's activity and response. But it can stand on an electric shock grid and also smell odors in an odor path here. And so this essentially allowed us to train the flies under the microscope. And so the first thing I did was give these 5-second odor pulses of 2 odors. These are the simple odorants we use for behavior. One I'm illustrating in purple and one I'm illustrating in green. And these are odors the flies have never smelled before at the time of the experiment. And so you can see that when the flies are naive to this odor, this output neuron responds really strongly. So the odor is being given during this little, it's hard to see, but there's like a gray box here of 5 seconds. So you get this really strong response to both odors, indicating that when the animal is naive to an odor, these synaptic connections are quite strong to the output neuron. And then I did a learning phase where I essentially paired short 5-second odor pulses with 1 electric shock pulse. And I did this, and then followed by 45 seconds later, I give the counter odor. which is another odor that that is not paired with electric shock. And so I did this 3 times. And at the end of that, I then tested the odors, the response to the 2 odors. And what I saw was quite striking. So the response is completely depressed to the odor that was paired with electric shock, whereas the other odor response is pretty much the same. And so I was quite surprised by how robust this phenotype was, and that the response is completely diminished to the paired odor, and it's very odor specific. And given that this output neuron is involved in the memory retrieval, leads us to, basically leads us to believe that this is a legitimate memory trace. So this is, some synaptic changes that are being laid out that are specific to the stimuli and that underlie the short-term memory of the fly. And so we then wanted to see what happens to this memory trace after we activate the dopamine neurons and do a couple of other scenarios. And so, sorry, just to back up one step. So this memory trace, just to kind of back up to the overall sort of behavioral circuit view, how this is underlying the behavioral avoidance of the odor. And so basically, what we think is happening here is now you specifically, because the shock is activating this specific set of dopamine neurons on this one side here, and not the other, we're affecting synapses that are synaptic input to this output neuron class that would normally drive an approach component. And so you're basically shunting that flow of information, but the flow of information still flows to the other side. And so you shift the balance towards avoidance. And presumably and other people have shown in other reward-based compartments, something similar is happening, but only on the other side of this circuit diagram here. So we wanted to see what happens to this memory trace when we activate the dopamine neurons. So we'll look at the forgetting aspect. And we did 3 different things. So the first thing we did was, basically the sort of baseline situation where we don't give any electric shock anymore, and sort of allow some time to pass and measure the response to odors. This is sort of the no shock condition. And what you can see is that the depression remains. It's relatively stable. It decays a little bit, and over time it will decay, completely by 1 hour or so. Because this is, I think we believe this is the short-term memory component that's being stored here. But during this period of time, it's relatively stable. But during this period of time, if we instead, in another group, we give an unpaired electric shock. So we give a shock that's not paired with either of the odors. And we do this, again, 3 times. What we saw was, I think, quite neat. So basically, we see no effect to this counter odor. It stays basically the same, the response to the counter odor. But the response to the previously paired odor, this depressed response becomes restored, essentially. It gets repotentiated back towards where it's more similar to the original naive response, indicating that activating these dopamine neurons, or this dopamine neuron, that it's involved during the encoding of the memory, but when you activate it afterwards, unpaired, you essentially can remove this or erode this memory trace. We also showed with data I'm not showing here, but we also showed using optogenetics. Instead of using shock, if you optogenetically activate this dopamine neuron, we see a similar thing, that you can pair it with odor, form a depression, and then unpaired, you can repotentiate it. And then finally, this last scenario I think is pretty neat, and it's basically looking at sort of like the idea of learning something new. What happens when the animal learns something new? And to do this, I simply, using the same set of stimuli, but we sort of rearrange it so that now the shock occurs with the counter odor, the odor that was previously not paired with shock. And so the shock is still unpaired with the previously depressed odor, right? So you still get this repotentiation happening, but you simultaneously get a depression to the other odor. And I think this is really neat because it's essentially like as if you've exchanged one memory trace for another. And it indicates that during new learning, 2 things are happening simultaneously. You're forming a new memory trace while you're eroding an old one. And so I think this gives a lot of sort of more sort of circuit level explanation for the sort of psychological idea of retroactive interference, where when you learn something new, it has a retroactive effect on previously learned information. And so basically that's how we understand how the dopamine neuron is sort of regulating the short-term memory, both the formation of it and forgetting. In my previous lab, a student named Martin Sabandal worked, he's a very promising student. He wanted to sort of follow up a little bit on how dopamine causes, or how dopamine regulates forgetting, but he wanted to look at, instead of short-term memory, he wanted to look at long-term memory. And so in flies, you can create long-term memory by doing something called spaced training. And so it's essentially the same odor paradigm, odor shock paradigm that I showed before, but you do it multiple times, 5 or more, and you space those by 15 minutes. That's why it's called spaced training. And when you do this, you produce long-term consolidated memory. And this memory it depends on protein synthesis, similar to mammalian systems. And in Martin's hands, his memory lasts for more than 14 days. He just stopped at 14 days because it's just too much work to keep them going for much longer. But the memory was pretty, I mean, it remains pretty stable for a long time. And so he wanted to look at how whether dopamine neurons have a similar role in forgetting. And the answer is yes and no. So there was a lot of interesting findings in this paper, but I kind of just sort of briefly go over a little bit. What he found was that the dopamine neuron I was looking at, gamma-2, actually had no effect on this type of memory. But he did find in a compartment that's in the vertical lobe of the mushroom bodies, this alpha-2, alpha-2 dopamine neuron compartment, that this dopamine neuron, if you activate it, or if external stimuli, for example, can actually activate this neuron. If this happens, prior to retrieval of the long-term memory, it does cause forgetting. So if you activate the dopamine neuron, flies will forget this consolidated memory, and also through this DOP1R2, similarly to the short-term memory, this R2 receptor seems to be involved in this type of forgetting, but the forgetting is transient. So if you wait long enough, the memory comes back. And that may make sense because it's protein synthesis dependent memory, and so it's more consolidated, and it appears that type of memory is somehow cannot be completely removed by this mechanism. And he collaborated with me to do some imaging. So he had me do some imaging to try to see in this alpha-2, alpha-2 output neuron. So he also showed that this output neuron here, alpha-2, alpha-2, was involved in retrieval of this long-term memory. And so we thought it was a good guess that there would be some kind of memory trace in the connections of the mushroom body to this output neuron. And so when I looked at that output neuron's responses doing very similar type of experiments that I just showed for the gamma-2 compartment, I did see a very strong memory trace there. And it was odor specific, and also it depended on protein synthesis. And so it likely is a memory trace that's underlying this memory, this long-lasting memory. But what was interesting is when we did activate the dopamine neurons and then look at the memory trace, it was not eroded. So for some reason, or mechanisms we don't completely understand, this consolidated memory trace, unlike the sort of labile short-term memory trace, this consolidated memory trace is resilient and resistant to that. But somehow, dopamine neuron signaling, we still don't understand, but somehow the signaling through this R2 receptor is doing something to block the retrieval of this memory trace. And that's still an open question in terms of how that's happening. But yeah, so dopamine does have a role in regulating forgetting of long-term memories, but it's in a different way, only transiently. So finally, I'd like to end the last part of my talk talking about sort of one of the projects that I'm very interested in right now, and it revolves around this dopamine receptor model. And so we have this model now where these two different dopamine receptors are being expressed in the mushroom body neurons, and you can see here, one of them is involved in learning and forming the memory. And in my data and other people's data from other compartments, that largely this memory is being encoded as a synaptic depression. And that, during forgetting, you get a kind of a repotentiation of the synaptic connection that's eroding the memory. And so we hypothesized that one receptor would be driving depression and the other receptor driving this potentiation. But before we got a chance to test that, Vanessa Ruta's lab at Rockefeller, they produced a study that showed very similar aspects to what I showed about the memory trace encoding and the depression in different compartments. But they also showed that one of these receptors, that this is indeed true, that R1 receptors was required for the depression and the R2 receptor was required for forgetting. And so we end up with this model where these two receptors are critical to the storage of the information, to forgetting of the information, for both the synaptic plasticity, different aspects of synaptic plasticity. And so we still know very little about what these receptors are doing downstream, what they interact with and how they're regulated. And so I really thought it was very important to really try to shine a light on what types of cellular systems and protein networks are involved involved with these receptors. And to do that, we tried in the past, in my previous lab, to use some antibodies to these receptors to try to purify them to do like a sort of traditional co-IP proteomics approach. But that was never very successful because the antibodies are rare and difficult, and we couldn't get very good purifications. But then basically I came across proximity labeling proteomics, and a lot of this has really been pushed forward by Alice Ting's lab at Stanford. But this involves proximity labeling as sort of a newer approach, and I think it's kind of an exciting evolution of proteomics. And so they're using these biotin ligases, which have been engineered in such a way that essentially the way they work is if you fuse them to your gene or your protein of interest, that they're mutated biotin ligases. And so if their free biotin is in the environment, they will convert this biotin into a highly reactive species of biotin that then is released. And it basically ends up forming a proximal or a cloud in proximity around the ligase of whatever protein you've fused it to. And what's nice about that is this biotin is a small molecule tag that covalently will bind to the proteins in proximity. And so you can then purify the interactors directly instead of having to try to purify the antibodies. Purify the GPCRs. And also, the biotin has a very high, specific and high affinity interaction with streptavidin reagents like beads that are commercially available. And so you can purify the interactors directly. And this labeling also is occurring in vivo. So you're kind of creating a history of potentially what's in proximity to the receptors and potentially interacting with the receptors. And so when I saw this, I really thought it really was a good solution to try to understand what these two receptors are doing. And so what I've done is I've made, using CRISPR knock-in approach, I've knocked in this turbo ligase, which is the sort of latest derivation that works in fruit flies. And so I've knocked it into the C-terminal tail of the receptors. It also has a V5 peptide tag. And so essentially I looked in the flies and essentially the knock-ins worked. So you end up with this turbo using a V5, anti-V5 antibody staining. You can see the receptors are expressed in the neurons across the brain and the neurons that they're supposed to be expressed in. The pattern looks intact, and the differences in their expression in the nervous system between the two receptors looks very similar. And if you feed the flies biotin supplemented food, for example, and stain the brain with a streptavidin fluorescent fluorophore, you can see in wild-type brains, there's very little endogenously biotin-related proteins in the neurons. But whenever you stain the R1 or R2 turbo flies, you can see that you get this strong biotinylation of proteins in the mushroom bodies and in the circuits where the receptors are. And so I think these flies are working, and right now I'm isolating using lots of fly brains or fly heads to essentially isolate the synaptosomal fraction to really look at the synaptic environments around these receptors. And I'm collaborating with Glenn Uhrig, and he's sort of expert in swath, and so we're going to try to do some swath mass spec on these samples to look at look for like protein networks around these receptors and maybe distinct between the receptors, and then obviously follow that up with targeted RNAi knockdown of candidates in the adult mushroom body neurons and look for a role in memory. And in the meantime, while I was making those flies, I decided to try this in cells, human cell lines, because we've shown previously that with a collaboration with Martin Mayenow's lab at Scripps, that you can express these Drosophila receptors in human cell lines and apply dopamine, and their G protein and secondary messenger signaling is preserved or similar to what it is in the fly neurons. And so I thought maybe we could learn something by trying it in these cell culture or cell lines as well. So I had these two constructs made of the two receptors and then a control that's just a plasma membrane targeted turbo. And so when I did two different independent mass spec experiments, I ended up with about 90 human proteins that were significantly more abundant around one receptor or the other compared to the control, or around both of them. And so that's what's kind of drawn out here. These are some of the proteins, and then the black lines are sort of the annotated predicted interaction between the proteins that came out of that. So we're interested in seeing if any of these are involved in learning or forgetting. And so I have been lucky enough to get a biology 499 student in the lab. It's my first student that's been in the lab, and she's been exceptional. She's got the behavior working in the lab already pretty quickly. She's very steady hand, so that's good. And so she's been, which is needed for the behavior. So I assigned her to try to do about 32 of the fly homologs genes that are homologs to these proteins for the R1 receptor and look at learning. So she's knocking these down in the mushroom bodies. And so this is some of her data real quick that she's gotten so far. She's not completely done yet, but so on the far left is the positive control. If you knock down the dopamine receptor, and we're measuring the difference from the control in terms of performance, because every experiment has an internal wild type control, and you can see if you knock down the dopamine receptor itself, R1, you get this really robust reduction in learning. And some of the genes she's tried actually seem to maybe have a phenotype that they're involved in learning. But I was surprised to find there was a lot of them that actually seem to do the opposite. They enhance the memory to learning. And so that may be interesting. So we're going to follow up on some of this downstream. So finally, I'd like to make my acknowledgements. I'd like to acknowledge my previous mentor, Ron Davis. He's been great. He's given me all the freedom to try all these, bring a lot of different things and approaches to the lab that I've carried with me here to Alberta. And I'd like to thank a couple of people that helped me with some of the stories. I didn't get to present all of their data, but Isak Cervantes Sandoval, who's now a PI at Georgetown, he helped me with the sleep study. I didn't really have much time to talk about, but he did some behavior. Anna Fan, who's some new faculty here as well. You saw her talk recently. She helped me with doing the structured illumination microscopy on the output neuron study and some behavior for that study. And then Martin Vandal did some of the behavior, or he did all the behavior for the long-term memory paper. Scripps proteomics score for the mass spec data. Then obviously I'd like to thank the Department of Biological Sciences for giving us the opportunity to come here and start our own labs and our own research, and so close to such a beautiful location. We're excited to be here, and also I speak for both of us in saying that we felt nothing but support and help since we got here, and we've been very pleasantly surprised at how supportive everybody is, and I look forward to meeting everyone as things get a little bit more relaxed over time, I guess, with vaccinations and whatnot. And obviously Halima Panu, the undergraduate student, to help me with looking at some of the Hexcel results. And I'm happy to take any questions. hopefully I didn't go over time too much.
Thank you, Jacob. I have to say, as your chair, I'm delighted to see you showing data from trainees already and building collaborations in the department. You've been here less than a year, and it's not been a normal year, so well done. And on top of all that, I know you've had grant proposals in and your CFI and all the rest of it. So anyway, pat on the back and thank you for giving a fascinating talk. I think we've got time for a couple of questions. If anybody wants to put their blue hand up or unmute and show your face or just speak up. There's a question in the chat there, Jacob, if you want, from Heather, I see.
Okay, yeah, so that question is about how do you know if fly sleeping or not? So yeah, a big part of it is motionlessness. There's a criteria the field uses, it's 5 minutes or more, and they haven't, well, actually it's a little more complicated than that, but in general we use like this little pathway that they walk back and forth, and there's a beam that they cross in the middle, and if they don't cross that beam in more than 5 minutes, we start counting sleep. And every minute after that they don't cross is a minute of sleep, for example. So that's kind of an operational definition, but in the beginning, people also notice that they will actually change their posture. They'll like reduce their body closer to the ground. And then also the other major ones are things like arousal thresholds. So if they've been sitting there and you try to give them a stimulus and it takes more stronger stimulus to get them to move than it does if they have only been at rest for a minute or so, then that's another indicator that they were sleeping, is the reduced arousal threshold. And then the other one is sort of the homeostatic aspect. So obviously if they sleep, a lot during the night, and if you prevent them from sleeping during the night, then they will be motionless a lot the next day, right? And presumably they're sleeping to compensate for their loss of sleep the night before. Hopefully that answers that.
Very good. I think Olav has a question next.
Yeah, Olav.
Yeah, I'm of course interested in the plasticity of the system and specifically maybe some changes in the life of the flies that don't necessarily relate to behavior, to learning, but that affect sort of this punishment versus reward structure in the mushroom bodies, and what do you think is sort of causing that? So let's say a female has, in one part of her life an aversion to male odors, but of course in other parts of her life she has to have an attractive. So do you have any idea whether that is that plasticity is coded for at the central regulator, or is it prior to that?
I mean, obviously I don't know, and I can't recall anyone measuring specifically or trying to study that specifically. I mean, I don't know of that type of work, but I guess conceptually that could be achieved in different ways based on the output neuron structure. I mean, yeah, so yeah, there'll be certain sensory information that comes with the male, obviously, that is going, so basically that's the point, right? So at some point, the sensory information is driving a different response than it does at a later point in life. And yeah, I mean, I could speculate there could be a lot of different ways that could be changed. I don't know if that would be plasticity in the sense that, well, it's plastic across the lifetime, yes. So, but it's more of an innate response that is changing, I guess, across. It's not associative in the sense that it's just something that's changing based on, the way it's evolved to change across the lifespan.
Right, but you wouldn't see cartridges in the mushroom body being reorganized or rewired, would you?
I don't think that has to happen. I don't know. I mean, it could, but I guess you could also change. I mean, the sensory information, like for example, pheromones and all that kind of stuff, those things are very sort of label-lined mushroom body neuron input, and so I guess those specific mushroom body neurons that respond to those kind of innate cues about like what's a male could just change their sort of synaptic connections to the output neurons in a certain way. Although I guess in this case, each one of them would be connected to two different classes of output neurons. So how they change that balance, I don't know. I mean, it would have to be axon specific, you know, to specific changes specifically to specific axons. Or you could change it at the dopamine neuron level, but I think it makes sense that it would be changes in the sensory information that has such a specific role, like pheromone type information. So I don't know. I mean, that's a good question, and it's a little outside of what I've thought about before, but yeah. So I know I didn't do a great job of answering it.
No, but you know, I'm just asking because I was asked about a mechanistic model for tachynynin sort of modulation of responsiveness, and I couldn't come up with anything, definitely not in the bees, so I was just curious about flies.
Well, I'll give it some thought because I think it's a pretty interesting concept. I'll think about it. I'll get back to you.
Sure, thank you.
Or something.
Very good. Are there any other questions? I don't see any. I got a blue hand from Maya.
Hi, Maya.
Hi, Jacob. Thanks so much for that super interesting talk. I just have a few quick questions, mostly about the links to behavior. First, with your novel odor, so this is just a novel odor that the flies themselves haven't smelled. It's not really a novel odor. It's not something they would not encounter in their life.
Yeah, let me think about carefully how to answer that. So I consider it not a novel odor because that this particular chemical is not, so they're simple chemical odorants. So this chemical has not been experienced by the flies before, meaning the collection of olfactory receptors that respond to that chemical have not been activated in that way before. Now, I don't know, it could have overlapping over olfactory receptor neuron activation to some other stuff in their environment that they do encounter before. And that could be possible, but these we basically view them as novel to the animal because they're small chemicals and we've never given them specifically to the flies.
So how would you know that they actually have receptors? Like, where did that thought process come from to try and get a novel odor? If you know what I mean?
Yeah, I mean, part of it is this has come practically from behavior. Like, I'm not exactly sure the history of why. There's like a set of like only a handful of odors that all the Drosophila behavior people use. I'm not exactly sure how they started using, why they started using some of these. But I know there's a lot of studies looking at olfactory receptor neuron physiological responses, electrophysiological responses to large panels of these odors. And so they do have some understanding of which olfactory receptor neurons respond to which odorants, and sort of the way that's encoded the next step into the antennal lobe. So some of that information is known. I guess we call it novel because we haven't given it to them before, and it shouldn't be in their environment.
So would you expect to see differences if you used a compound that you know is super important to behavior?
Yeah. Yeah, and some people have done that. So there is like apple cider vinegar, for example, is really powerful, like innate attractive odorant to the flies because they like that helps them figure out where rotting fruit is, for example. So that has a very strong response, but that also has plasticity in it. So people have shown you can, for example, like in the output neuron I'm looking at, for example, if you were to give them that odorant, that output neuron would have a really strong response to it, but you could still depress it. If you paired that odorant with an electric shock, they're still able to block that, to modify that experience. So I'm sure it changes though. I'm sure it probably changes how much training you would have to do to block it to appeal, and also maybe how stable that version is. They may go back to liking it quite quickly. I don't know. I don't. That's an interesting question, and I haven't seen anyone really try to probe like whether it's different than a novel odor in terms of the plasticity.
Just two quick more questions, Dave. I know I see you. One is so many of these like behaviors are either like multimodal responses, including visual cues and chemical cues. And so I'm wondering if anyone's looked at sort of whether they can disrupt different neurons responding to those different modalities of cues. And then the second question is, a lot of the behavioral work in insects, some of it is flight gated. And so you wouldn't expect to see a response unless you let them fly. And so I'm wondering also about whether other behaviors, other assays are used in this kind of work.
Yeah, so the first question, basically you were asking, oh, multimodal, like other sensory information. So all I can say about that is there's been some work with visual information. So they know there's a specific set of mushroom body neurons that respond to visual information, and they know what's upstream of that, like what's bringing that visual information in. And the dopamine neurons still play the same function that they have, that I've shown. So they'll still, alter these specific set of mushroom body neurons responses. And so you can actually train them to avoid a color or even other things by activating the same sort of aversive dopamine neurons, for example. And so as far as multimodal, I mean, there's also some data that there's like tactile information comes in. And so I'm sure a lot of different information is getting funneled into the mushroom bodies. It's less studied than the olfactory, so it's not as mature as a study or a field yet as in the fly. But now the second question is about flying. And so there is another assay that is used relatively frequently. I think it's a lot harder to do, so it's not used very often, but it's a flight simulator. So there they tether a fly to a wire, and the wire is like magnetically balanced in a magnetic field, and they use like an infrared laser to like burn the fly as a reinforcer. And there's like a arena around the fly that has like, they can put like classically what's done is blue and green colors, or a T shape or an upside down T, and you can actually train them to like avoid a particular T versus the other T, or a color versus the other color in flight. And as far as anatomically, that's not as well worked out, but I think it still involves the mushroom bodies, the dopamine neurons. It's unclear the difference between, like differences between how they're behaving when they're walking around to odors versus flying. I think that's, I'm not aware of that, of a study sort of trying to parse those differences, but I'm sure there are differences.
Thanks a lot.
No problem.
A literal flying circus.
Yeah. Yeah.
We have time for one more. And I just saw congratulations from Ted in the chat. Anybody else with a question for Jacob? Yeah, so on behalf of everybody, thank you, Jacob. And yeah, I see some sentiments in there about we're really looking forward to meeting you in person, those of us who haven't. So yeah, hopefully that will come soon. So with that, I will wrap things up, and I will simply give you, virtually hand you your highly coveted Inspired Discovery for a Better World, University of Alberta Biological Sciences water bottle, which will be found in your mailbox. Do you know you have a mailbox?
I do. I checked it.
Good. Okay, good. All right, so it should be there. And the last thing I'd like to say is that we have one more chairs lecture this academic year, which will be Dr. Casey Fowler on April 8th, and I look forward to seeing you all there. And thank you again, Jacob. That was really enlightening.
Thanks.
Well done. Everybody, please enjoy yourselves and stay safe, and I hope to see you all soon. Take care.